Hi there. Good afternoon or good morning or good evening, wherever you are. And welcome to this most recent collaborative webinar. Today, Flow Cytometry has got together with Curiox to deliver a webinar for you today. Before we get on to that, just a brief reminder of what Flow Cytometry UK is. It's an umbrella organisation for all of the local flow groups in the UK. We were formed in 2006. If you're not on our mailing list, please visit flowcytometryuk.org, where you can add yourself to that and to get information about upcoming meetings and courses and webinars like this. We also run a scientific meeting every year. This year that will be in November. Looks like it'll be a virtual meeting, but hopefully next year at the University of Birmingham we can get back to our face-to-face meetings. That should be in July next year, and details of those meetings are on the Royal Microscopical Society website. But what we decided we needed to do during this time is have a conduit for companies to talk to us about things that are relevant to us in the cytometry field. So that's the idea behind this whole series of webinars, because without any exhibitions and meetings, it's very difficult for companies to speak to us. And of course, we still need to find out what's going on there. So today we're actually going to be looking at something that's maybe a little bit tangential to flow cytometry at first glance, but I do a lot of teaching in flow cytometry, and one of the things I try to hammer home is that your experiment is only ever going to be as good as your sample preparation. So as well as thinking about fluorochromes and panel design, you have to get that single cell suspension, and those cells need to be happy, they need to be viable, and we need to have as many of them as we can get. So sample preparation is extremely important. So we're going to hear two talks from Curiox today, one from Noma Ngwenya, who's the application specialist for Europe and the Middle East, and from Christophe Ibel, who is at Charles River Laboratories and a user of the equipment. And what we'll do is we'll have these two talks back to back, and then we'll deal with any questions that have come in towards the end of the hour. So without any further ado, I will hand over control to Noma, who can tell us a little bit about the Curiox system. The floor is yours, Noma.
Thank you, Derek. Hello, everyone. My name is Noma, as Derek just introduced. Today, I'll be talking on the Laminar Wash technology. And just to echo on the message that Derek really just presented about how key the sample prep process is. And that, as you can see, is key to getting much better, cleaner data, and just to make sure all your cells are happy. So current protocols, when you're doing flow cytometry, or single cell genomics, the main roadblock that we see is here with the sample prep using the outdated centrifuge method. So quite a few problems are associated with this method. It's quite difficult to automate this with different systems and liquid handling robotics. You get a lot of inter-user variability. Some people are more trained and more skilled than others, and it does cause a lot of disruption to your cells. So you do get changes in the morphology, and you do lose quite a lot of cells as well. So this is where the Laminar Wash comes into, where we give you a nice option for automation. It's fast, you get consistent results, and it's much more gentler on your cells. So your cells are much happier in the environment, and you get to see all the little morphology really well. So what is the Laminar Wash technology? So we've got a unique plate design that doesn't have any walls. So I know everybody always panics and wonders, how can you do anything without the walls? But we've got this hydrophilic coating within the wells and a hydrophobic coating between the wells. Now this holds your droplets. So as you can see in the video here, you add your sample that contains your cells and your antibodies in a suspension. You add up to 80 microliters into the wells. This then you allow it for the cells to settle using gravity, so they gently settle to the bottom of the plate. And any unbound antibodies or any unbound debris, anything floating, will then just float to the top of your droplet and doesn't affect any of the cells that are sitting at the bottom of the well. You then load on your plate onto our HT system, customize the flow rate, which means how fast the system washes the cells, and you input the number of washes. The head then goes down. You get each well gets given 2 nozzles, one dispensing, one aspirating, and you gently washes your cells. So it dispenses some buffer. It creates a nice laminar flow at the top and gently washes away all the debris and any unbound antibodies at the top, while it's undisturbing these cells that are settled at the bottom of the well. So you can see with some data here, which just shows the impact of centrifugation on the morphology of the cells and also on the staining index fluorescent signal that you get, comparing it with our laminar wash system. So you can see here in the centrifugation, the cell quality is quite poor. You can see the cells are looking more crushed. There's quite fewer cells as well, and the fluorescent signal is weaker. When we compare here with the laminar wash, you can see your cells, this fluorescent signal is much higher. You can see the morphology of them as well, and we have also retained much more cells compared with the centrifugation method. And here, this is just a nice little graph that can represent how we can retain much more cells compared with the centrifugation, particularly here at the lower end. And this is because it's when you're working with a pellet and you're working with quite low cell numbers, it's quite hard to guess where the pellet is. So you do end up then washing and getting rid of some of your sample, whereas the laminar wash removes that guesswork for you. So when we just focus on the methodology of the system and how a simple protocol would work, we can see here at the beginning, you would stain your samples with a viability stain or any antibody stain here. And each well can accommodate up to 10 million cells. Ideal retention, and we work out, is between the 1 to 5 million, but you can do less than that as well. We would do a wash sequence. So here, if you did 2 rounds of washing, that would take approximately 10 minutes, excluding you doing any of the flicking. And here on our system, when we set that up at a 10 microliters per second flow rate, you can see that would take 1.5 minutes to just go through that process, where you would have done 10 plus minutes. And our settling time is approximately 30 minutes, and you can combine this with the staining step. This 30 minutes is quite dependent on the density of the cells as well. So for example, if you're using yeast cells, these are much larger. They tend to settle down on the plate much faster than those that are slightly smaller. And we do work, the plate also works well with any cells that are any adherent cells. And you can directly read from our plates onto most cytometers with the addition of this accessory, which is our big volume adapter. And that just increases the capacity of the well to then directly load onto cytometers. And with using our laminar wash, you can see we get improved staining index, resolution, much better population separation of cells, and a much better MFI. So here I've just got a video which just shows you in real time the differences of running a centrifuge method with our laminar wash method. You can see just doing all of this simple prep would take you approximately 25 minutes. Whereas using our HT system, the whole process would take you 5 minutes. So that's great news for you if you're in the lab, because that gives you more time to go and do other analyzing work, or as I used to do, go on a nice tea break. So here you can also see our system fits very well in biosafety cabinets with the current state of events with the COVID research. This is much more critical to make sample prep safer. So you can see it sits very nicely into a fume hood, doesn't take up much space. You can see our FAS team, when we come on site, we do wear masks as well. And we have no production of aerosols. So it's protecting you and everything around you from anything. So we have broad uses of our system. Today we've got flow cytometry, but we do work with single cell sequencing. We work with automated workflows, vaccine development, tumor microenvironment, to name a few. And my next slide here just shows some of the current customers that are currently using our system, all with different areas that they focus on. And today we do have our guest speaker from Charles River as well. And here we just got a couple of testimonials from our customer, but the background message that we get is that you see that we have better retention, we have less data variation, and within operators as well, because it's much more reproducible. You get tighter control over your populations and much better data quality. So we offer 3 different SIS platforms with our Laminar Wash. We have our low throughput system here, which is our Mini 1000, up to our middle-sized one, the HT 2000, and our higher throughput, our Auto 1000, which is a simple complete walk-away system that allows you to do the whole sample prep procedure within the Hamilton Nimbus. As well, additionally, with our HT 2000 system, you can see we can integrate with other robotics. So here we've got an integration with the PAA S-Lab, which would just simply load on the plate for you and will begin the washing. And with this, you can run 10 plates in approximately 20 minutes. So we would also provide automation licensing key and work with the robotics company in making sure you can get your system up and running and integrated with that robotic handler. And we do offer this extra accessory with the buffer exchanger, which allows you to connect up to 6 different buffers to the system, particularly great for assays where you're constantly switching with different buffers. And this is just an overview of our Auto 1000 simple walkaway system. So here on our left is our graphical user interface, which allows you to change the different parameters associated with the assay. And here you can just see a bird's eye view of how the system is set up. So in this portion here, you would have the Hamilton Nimbus. And then here is where your Laminar Wash system will sit, with all the associated plates, and we do have a temperature controlled Peltier for all your incubation steps. So we do know those assays that need to be temperature monitored, so we do have that option as well. So here is just a really quick video which just shows you the process of how the simple automated system works. So it runs 2 plates in series. Oh, sorry. It's gone fast. Sorry. So it runs 2 plates in series. It will pipette the sample for you. It will add on all your antibodies and your viability dyes. It would also perform any fixed perm as well. Loads on the plate for incubation. And it will also then load on the plate to be washed. So here I'm just going to talk about some data with some of our partners that will currently have our system. So here I've got some data from Cerevance. This is a private clinical stage pharmaceutical company which focuses on brain disease. So they first approached the Laminar us with Curiox because they wanted to retain much more nuclei for their downstream processing. And from our work that we've done, we've also managed to increase the nuclei retention on average between a 1 to 3 fold. they also noticed that they're using 10 times less antibodies, so they're saving costs on any of those expensive antibodies. We've got comparable staining index, and our laminar wash increases the percentage of the single nuclei, so their cells of interest. So I've got some data here, which just demonstrates how the laminar wash has improved their data quality. So here, one of our tests was performed on 100,000 cells on a single stain conjugated antibody. So we can see just from looking at this antibody A, we can see already the laminar wash had much more cells retained compared to the centrifugation. Our staining index is comparable, even with them using 10 times less antibody. So then they're saving on the cost as mentioned. And here you can just see the values of the single nuclei, their cells of interest. You can see here they retained 36,000 with the centrifuge, whereas here with our laminar wash, we saved approximately 34,000. So that's double their cell retention for their nuclei. And here we just got a nice little summary, which gives you a more summary of the different tests that we did with the different starting cell numbers, starting from our lowest at 50,000 up to 500,000. You can see across the board, we are very consistent in the testing. We can see we have this exactly much higher retention than centrifugation. Whereas here with the centrifugation, you can see there's quite a lot of variability between the testing, whereas we are quite consistent. And this just shows the fold increase, which just shows you a similar picture. So just to round off on my presentation is our recent white paper that we've released in collaboration with Charles River, with Christoph as well, who'll be going into a lot more detail of some of the work that they've done with improving their sample preparation with TILs. So they wanted to use the laminar wash system because they wanted to improve the recovery of any viable TILs from dissociated tumors. They wanted to reduce any inter-operator variation as well. And they wanted to shorten labor-intensive hands-on washing steps associated with centrifugation. So here I've just got some of the data from the white paper. You can see between the tumor-naive cells and cells that are treated with this APD1, both of these show you a similar picture that laminar wash has higher viability of cells. You do have differences between different users, and you do have much lower viability for both of the TIL populations. And then here from the flow plot, you can see that with laminar wash, it's much, much cleaner and it's much easier to gate around the population of interest. When you compare here with centrifugation, it's quite hard to gate and find that population you want. And you can see that we have got a higher increase in the viability. So to summarize, the laminar wash improves on-time savings. It removes that hands-on time, so you have much more capability to walk away, focus on other things, and come back. It's consistent. It removes that operator variability. You can automate the whole process. Great for biosafety, particularly with the time and the events that were happening at the moment. And when we're looking more on your cells, you've got the cells remain in their native state, so they're much happier. You can see the morphology better. You get higher viability and retention. Much cleaner data because you can remove all of that, gently remove that debris, and you get enhanced cell washing, so you get a reduced staining background and clearer resolution. So I'll just pass over to Christoph now, who will just speak about his experiences with the laminar wash in more detail.
So hello everyone, thank you Noma, and thank you for allowing me to share now a bit of how we at Charles River applied the Curiox laminar wash technology towards standardizing sample preparation workflows for flow cytometry. this is for us an ongoing learning and adaptation process in a discovery research environment. Therefore, I want to begin by giving a concise overview of how we are set up at our tumor model group in Worcester, Massachusetts, and then how we removed centrifugation from cell-based sample preparation and immunostaining protocols to streamline the flow cytometry workflow. Using side-by-side comparisons of centrifuged and non-centrifuged cell suspensions processed from the same source, I want to highlight that it is worth challenging the status quo with centrifugation as the go-to instrumentation when handling cells. Definitely, it turned out to be worth in our line of work. The simple examples presented will be focused on dissociated tumor suspensions, since oncology and immuno-oncology studies are our group's forte. Finally, I'm going to summarize the major benefits the Curex washing technology can offer towards standardizing sample processing. Our flow cytometry operations are integrated with the in vivo pharmacology group at our discovery facility located in Worcester, Massachusetts. And the major intent was to synergize sponsored mouse oncology studies with PD immune endpoint readouts in a strictly non-GLP environment. This setup made sense from a pre-analytical point of view, since tumor samples can be collected and seamlessly processed in close proximity to the vivarium without delay. Essentially, our line of work attempts to answer two questions. Is an experimental anti-cancer immunotherapy efficacious or not, and why? In addressing the why, exploring the tumor microenvironment comes into play by profiling the composition of immune cells and subsets and changes therein. However, the answer may sometimes be not as straightforward as hoped if there are discrepancies between efficacy and tumor infiltrating lymphocytes or TIL datasets, as we also try to better understand the optimal time point in an efficacy study to investigate immune cell infiltration into the tumors. Apart from the flow cytometric analysis of study tissue samples, we also enable humanized mouse model use for preclinical research. And for all these services we offer, we rely on Attune NXT flow cytometers configured with 4 lasers for 14 color detection. And as some of you may know, or many of you who use the same system, the noteworthy technical detail is the acoustic assisted hydrodynamic focusing, which pre-aligns cells in suspension more accurately and faster before passing the interrogation point. We all know many factors influence flow cytometry data output. It is deservedly described as information rich, and common sources of variability include the way instruments are set up to record samples and how these data sets are subsequently analyzed. Technical solutions are already offered to address inconsistencies that can be introduced at each step of the flow cytometry workflow, all of which are intent on minimizing or eliminating human error and bias. Many of us eventually have heard the phrase garbage in, garbage out to emphasize the relevance of how well samples should be prepared. On this very relevance, we have been focusing in our line of work, and that is why we are exploring a next generation technology like the one we are presenting today. Building up on what Noma was already presenting, when suspension cells are processed without the centrifuge, this not only requires a different way of sample handling, but also numerous considerations for workflow standardization, as well as factors affecting assay performance in this novel setup, some of which I have listed here. Among the first variables to optimize is the initial settling time for the cell sample type typically processed in your laboratory. In terms of how gravity is used, this step presents the major difference to traditional pelleting and plate flicking using centrifugation. As rule of thumb, at least 30 to 40 minutes is a good starting point for commonly dissociated tissue samples, including spleen, lymph node, liver, lung, and tumors. Of note, we did not manage to work with platelets in this setup, which would have been advantageous since platelets get activated by centrifugation. The number of wash cycles between incubation steps can influence staining results. Besides that, it is important to figure out whether stain samples can be directly acquired from a 96-well Curiox plate for flow cytometric readout, since manual transfer to a reading plate, as we did before, introduces variation again. As you can see here, the centrifuge-less cell preparation platform is enabled by a wall-less plate. Like traditional plates, it consists of 96 wells with an area of hydrophilic spots surrounded by a hydrophobic surface, which functions as a virtual wall that separates each spot. For processing, we aliquot 70 microliter droplets of suspensions with typically 1 to 2 million cells, but theoretically, you could deposit just a single cell. The washer then enables a laminar flow in each well, fully automated as programmed by an operator. And for us, instrument maintenance has turned out to be a particular area where improvement was needed. Since we process many different dissociated tissue samples often at once, we needed to counteract the risk of nozzle clogging and increase debris deposition in the platform by more preventative and regular maintenance protocols. In this figure here, you can see where debris from tumor sample processing typically gets deposited, requiring these parts to be exchanged. These examples are merely for illustration, since proper customized maintenance can minimize this issue. With the laminar wash technique, cells in suspension undergo gentle wash cycles without added stress, allowing a higher retention of cells for staining. For high throughput applications, this setup supports achieving consistency by reducing variation from manual handling. I like to compare this approach to gold panning, with immune cells being the gold dust or nuggets hidden in plenty of debris in a dissociated tumor sample. And in these figures, you can see that when the same spleen and tumor samples collected from naive and CD26 challenged mice are simultaneously processed by centrifugation and laminar wash, more viable cells are retained for immunophenotyping by the latter one, irrespective of the initial number of cells plated. We evaluated technical replicates of 3, starting with 1, 2, and 5 million cells. And we believe that the more effective washing away of floating debris from the pre-settled cells in the laminar wash plate, particularly in dissociated tumor samples, contribute to the increased recovery. Less manual handling also improves the overall CVs, as you can see here. And in case of the laminar wash platform, those we typically can keep below 10% for most cell types we tend to process. To also compare the laminar wash system with other TIL purification techniques, prior to immunophenotyping, we compared the debris removal solution procedure and our laminar wash protocol side by side on preparing MC38 dissociated tumor samples. Just for information, the debris removal solution relies on density gradient centrifugation in 2 steps. And here are the dot plots of MC38 TIL samples processed and later stained with CD45 on only centrifuge samples. In this case, debris removal increased the frequency of CD45 positive cells, but lowered cell viability when compared to the same sample process without prior debris removal. And here are the dot plots of MC38 TIL samples processed and stained with CD45 in parallel, using laminar washing with and without debris removal. In direct comparison, and when comparing to the technical replicate set that was only centrifuged, it is clear that laminar washing by itself, if you just look at this dot plot here, by itself yields the best results in terms of cell recovery, viability, and overall consistency. If combined again with debris removal, these established laminar wash benefits tend to get lost. And these examples are representative of a trend we have observed for similar side-by-side comparisons. So besides that, we incubated MC38 TIL samples processed from naive C57 mice with annexin 5 and propedium iodide to understand whether there is a difference in the number of apoptotic cells depending on the processing method. Not just in this experiment, but generally, we noticed that the number of cells undergoing membrane rupture tends to be higher in centrifuge samples than in those processed by laminar washing. A result not too surprising, given that we know centrifugation adds mechanical stress on cells. Associated with our immune profiling of infiltrating lymphocytes, we increasingly face questions around metabolic aspects of the tumor microenvironment, for example, in tumor-associated macrophages. So generally, we were curious and received requests to understand whether the traditional sample preparation by centrifugation could render cells more hypoxic. We found that this appears not to be the case when comparing splenocytes collected from a naive biopsy mouse processed side by side, as you can see here. Splenocytes were plated and processed by centrifugation and laminar washing in technical replicates of 5 for each method. We used the commercially available hypoxia green reagent to check for oxygen levels in live cells. Briefly, this probe can permeate the membrane and creates a fluorescent signal from released rhodamine in a low oxygen environment. And as positive control, we incubated splenocytes at 37 degrees for 2 hours at 1% O2 levels, which is not depicted here. But here we found no indication that cells turn hypoxic by repeated centrifugation as seen in these plots. Cells remain normoxic regardless of the processing method. Next, I want to share an issue we encountered with one of our study sponsors. In tumor samples processed from MC38 and SAL-N mice, we observed a clear and in some cases large CD3, CD11b double positive cell population. Our study sponsor did a more high-level non-biased data analysis in-house, and from this gating, CD11b positive cells were identified that are both CD3 negative and CD3 positive. So from both sides, there was a great interest to understand whether this is a real subset within the TIL population or merely a staining artifact. We suspected actually the latter, as in our troubleshooting, we found no indication of a compensation or FMO issue, no carryover, or cross-contamination of stained samples. And when we looked into the literature, there were suggestions that the CD3 positive macrophage population may exist, although at this point it appears not to be well characterized. From other discussions, I gathered that in attempts to better understand the biology of macrophage populations, these TCR expressing macrophages may occur in context of inflammation, but the question still remains if this would apply to the tumor microenvironment as well. So absolutely no clear picture here and no easy answer. So in the end, we suspected that most likely we are dealing with a staining artifact. Therefore, when we performed antibody titrations and also did side-by-side comparisons of centrifuged and non-centrifuged samples, we noticed that the double positive population can be resolved by retitration of the CD3 and CD11b antibodies. Though this issue did not occur before, we realized that we can and need to retitrate antibodies even more so in the smaller staining volume used on the laminar wash plate. So in this case, we actually can go down with CD11b beyond this dilution factor down to 1 to 1000, while with CD3 we discussed keeping the original 1 to 100 dilution. So once we worked all this out, we worked again with the retitrated dilutions and with the laminar wash method in follow-up TIL immunophenotyping experiments. We did not find the CD3, CD11b double positive population again. And when we also directly compared centrifuged and non-centrifuged sample sets across the titration range tested, we could confirm once more the increased cell recovery and increased viability the laminar wash technique offers. So there was no apparent influence of the washing technique on the occurrence of this artifactual double positive immune population. I can only speculate here whether a different way of sample preparation, like the one offered by laminar washing for downstream analysis like flow cytometry, could one day help us uncover an immune cells subset that we could not stain for before because it simply got lost during centrifugation. So since we adopted laminar wash technology, it helped us streamlining our sample preparation procedure for tumor microenvironment characterization by flow cytometry after mouse tumor studies and in our group. There are two benefits I want to highlight for that matter. Through automation, we can simplify washing steps in immunostaining protocols and save time needed to perform those. Particularly for high throughput shared resource labs, this option should be explored when efforts towards consistency are made. By minimizing variabilities in cell-based sample preparation typically introduced by human error, simple human error by the way various operators handle cell suspensions, we observed better staining results in terms of reproducibility and overall data quality, a notion which we frequently get by our research partners and collaborators when processing their samples, their study samples. This novel way of washing by laminar flow keeps cells unperturbed as much as possible throughout processing, which results in improved recovery of healthy, viable, and well-defined tumor-infiltrating immune cell populations for more downstream analyses like multicolor flow cytometry. And adapting our standard immunophenotyping to the laminar wash standard and reduce staining volume has allowed and still requires us now on a case-by-case basis to retitrate antibody conjugates, resulting in further reduced reagent usage and cost reduction. I also want to emphasize that there is no one-size-fits-all approach when utilizing laminar wash technology. There are key variables that need to be optimized, from the number of wash cycles and settling times to sample handling post-staining. Fortunately, we could configure our autoassembler to support direct acquisition from the Curiox wall-less 96 well plate on our attuned flow cytometer. Lastly, as an add-on benefit, we now can train new qualified users faster, actually faster on operating the laminar wash platform for suspension cell sample preparation. I hope that after this glimpse into our learning experiences, you too got curious about this next generation type of washing technology, how it can support cell-based assay standardization and fit in with applications other than standard immunophenotyping using flow cytometry. For example, RNA sequencing, cancer organoid investigations, or single cell omics, just to name a few. And with that said, thank you everyone for tuning in today, and thank you to the hosts for providing this forum.
Excellent. Thank you, Christoph, and Anoma as well. My camera there, you can see me. There are a few questions that have come in, so if you've got a few minutes, we should be able to get through those. Some of them are technical, I think, and some of them are more to do with the procedure in there. So the first one is about the buffer exchanger on the machine. Is that something that's mandatory when you're prepping your cells for flow cytometry, or is it something that's just good to have? Is it an option?
It's a customizable option. It's not mandatory, so it's much more simpler. It's much easier to use because it's it just automates the whole process for you. However, it's not something that you definitely need to add on.
Okay. And the plates that you're using, are they reusable? And if so, how many times can you reuse them?
They're reusable in a sense that you can reuse the wells that you haven't actually put cells on. So the ones that you would have used for the test, as the system washes the whole plate in one go, you would just mark off the ones that you've put sample in, give it a wash and rinse through, let that dry off, and you'll be then able to use the remaining unused wells at a later stage. So you can just keep reusing those unused ones until you fill up the plate, essentially.
Okay. I suppose this is one for Christoph in terms of the way you've set up your automation in your lab. Have you considered actually fully automating this, or moving between the Curiox prep system and then the Attune?
Yes, absolutely. I mean, the subject is actually currently under discussion. So we want to actually move from this semi-automated way the operations is set up to a fully automated version like this, what Noma presented, the turn away or the walk away and the turnkey auto platform, which would not, in our case here, I think we want to go step by step. So right now, the way the operations are set up, so we try to formalize this more now in terms of, from an SOP perspective, because QA is on our heels here. And then once, this is all set up, then actually have a discussion or finish the discussion on when we can introduce this full automated system for the very same sample types we are processing. So midterm discussion would, so we are looking probably into next year, of course, I mean, with some of the procurement, that goes around it. But absolutely, in one sentence, we want to fully automate this process for at least some standard assays we are running, and then go maybe beyond that and see if we can adapt this also with our friends from Curiox for some more exotic assays, which we typically wouldn't run so often.
Okay, great, thank you. This is another one possibly for Noma, and I think one of our delegates maybe have missed this about the debris removal process. So can you use that? I mean, is that something that's built into the system?
So yes, so our system is very good at removing debris in your sample. So when you load on your sample onto the wells and the cells settle, the debris tend to float at the top anyway because they don't have that much of a density. So then the system just gently washes them off, and it doesn't disturb your cells that are settled at the bottom of the plate. So it's very efficient in removing debris and any background artifacts.
Excellent. And the plates you use, are they completely proprietary, or they come from anywhere else as well?
They're from us. They have their specific plates.
Okay. Actually, one thing that I may have missed there is there a sort of limit in the size of the particle, an upper and lower limit in the size of the particle, and does that influence? Does the size of the particle influence the settling time, for example? Presumably it does.
Yes, so the bigger the cell and the density, the quicker that it will settle. For example, if you're using cells like Saccharomyces, those are quite larger and they would settle much quicker. Within 10-15 minutes, you'd get very efficient settling. Much more smaller, much less starting material, you would be working at minimum 30 to 40 minutes settling time.
Okay. And there's a question here about the applications. So would you say the applications would be sort of fairly broad for any sort of flow cytometric technique?
Yes.
In terms of cell type, I suppose, that you're using?
Yes. Yeah, that's a very good question. Yes, we do have a quite variety of customers using our system in a very broad range of flow cytometric assays and platforms.
Probably another one for Christoph as well, because you mentioned training of users. This is something that I suppose is very important to those of us who run core facilities or shared resource labs. And how easy is it to get people up and running and trained on the system?
So I have there a good example from a research associate, which actually expressed an initial interest in working with flow cytometry, sample preparation, and learning the whole process. So he is an example of a user who had no prior knowledge, and it took us pretty much 1 month to get him up and running with this sample preparation procedure. So going through our typical, SOPs for training, but he was, so somebody with no prior knowledge, as I said, within 1 month, he was fully proficient in such a way that I can now comfortably walk away and he handles everything from start to finish. And with very good outcome. No, no samples are lost, no artifacts, no questionable data quality. So 4 weeks for somebody who had pretty much, had no idea about all of this before, I think is a pretty good start. And as I say, then the next ones are already in line, and I don't know, maybe you can, I learned this within, within 1 week in a demonstration with Curiox. So I think probably, if depending on your prior knowledge, so say between 1 week and 4 weeks on new users, because it's really so easy to operate. And once you know how to, to set your mind from, the centrifugation handling of samples to this new setting, I think then you can pretty much go with that, seamlessly and easy.
Excellent. So maybe a little bit quicker than training somebody to use a high-end flow cytometer.
Definitely, definitely.
There's one more practical question here about the plates, the wall-less plates that you use. Do you have to? Is there any differences in the way those are handled compared with the standard microplate?
You have to be slightly more careful with the plates because they don't have the wall to prevent if you accidentally shake the plate with vigorous action, it can cause a bit of well merging, but that also can be how much is actually on the well dependent. So if you've got a quite low volume, you're unlikely for the wells to merge. So it does take a bit of training and getting used to the handling of the plate, but it's not a huge difference. It's just, you just need to be able just that 5-10% more careful.
Okay. Thank you. I think we've come to the end of the submitted questions there. So thanks everybody for those. I think those are a couple of great presentations and some good questions and answers there, and very interesting technology, I think, for those of us who are trying to deal with optimized sample preparations when we come to the flow cytometer. So obviously, if there are any questions that people think about afterwards, the contact details should be available on the screen, or I'm sure you can reach out to Curiox or your local Curiox representative to help answer those. So I suppose it just remains for me to say thank you all for coming. Keep an eye on the Flow Cytometry UK website or Twitter feed for upcoming webinars and meetings. And we hope to see you all in real life in the very near future. So once again, thank you all for coming.